want to start with some basic principles just to remind us all of things that you might not remember if you weren't the fortunate recipient of a UCSD education as I was. Remember that our bodies are built of cells, and cells in a way are like small businesses in our communities. They're each, as we say, specialized to have different jobs. So different cells in our body make different products or provide different services. And you know some examples of that already, right? So cells in the pancreas, for example, one of their jobs is to make insulin so you can digest food. Cells in your brain, their job is to transmit electrical signals so that we can think and move and so on. Cells of the heart, their job is to contract to pump blood so that our tissues can get oxygen. Now, what's not so often realized is that many of the diseases that afflict us are caused by one sort or another of cellular breakdown. And this really happens in a couple of major ways. One major way is where the cells are still alive, but they're damaged in some way. And heart failure is a good example of that, where the cells in the heart that contract to pump blood, they're still alive, but they're damaged. They've undergone some degenerative changes so that they don't contract as well anymore, and they don't pump blood as well. And as a result, you get the circulatory and other symptoms of heart failure. Second category of disease is where cells actually die and are gone. Type 1 or juvenile diabetes is an example of that, where the beta cells in the pancreas have died and they're gone, and so they can't provide insulin anymore. Or in Parkinson's disease, where special cells in the brain that are involved in the control of movement die and are gone. Now, if you look at the long list of the more common diseases that afflict us, what you find is that many of them are associated with one sort or another at their roots with one or the other of these types of cell damage or cell loss. And you can see the list, it's pretty tremendous. We mentioned heart disease and diabetes, spinal cord injury, Alzheimer's disease, Parkinson's, blindness, muscular dystrophy, many kinds of kidney and liver disease are ultimately cellular diseases of one sort or another. Now, if we ask the question, why don't we have better medical therapies for these diseases than we currently have? I mean, the ones we have are often quite good, but they could be better, I think, in many cases. What are the bottlenecks? What keeps us from more rapidly devising better therapies for these diseases? And I think there are several pretty obvious reasons. First is, for treatment at least, in many cases, we need sources of cells or organs for transplant. So if you need a new heart, if you need a new kidney, if you need a bone marrow transplant for leukemia or lymphoma or sickle cell disease or something like that, sometimes we can find donor tissue, sometimes we can't. In fact, often we can't. And in many cases, for example, for type 1 diabetes, the source material is incredibly limited for transplant. We just don't have enough, and in some diseases we don't even really know of a good source. Second issue, and an issue that drives, in my view, the cost of drugs, is that we don't do as good a job as we might in figuring out which drugs should be tested in human clinical trials. And there are two real reasons for that. Remember, a lot of our drugs, before we test them in humans, get tested in animals. And it's only after there's been successful animal testing, where successful means they didn't kill the animals, and they got better from their disease. And the disease is often a version of the human disease. But remember, we are not mice. We're similar in some ways, but we're not identical. You may notice that. And the unpleasant surprises that frequently happen in clinical trials are unexpected toxicities in people, often liver or other toxicity. And in fact, we got a recent unpleasant surprise with Vioxx, where, in long-term use in humans, a toxicity that had not surfaced in animals and didn't even surface in short-term clinical trials became evident. And so prioritizing drugs so that those that are not going to eventually be toxic we don't even put into clinical trials, that would be good, right? And remember, clinical trials are really expensive. You're looking at 200 to 500 million a crack per drug. Yeah, that's real money. And then the other problem is sometimes things that cure mice don't cure people. They don't work as well in people, right? And again, that's a big issue, and you'd like to weed those out before they even get into humans. Third issue is for some diseases that we would like to treat, we don't actually understand the disease. So we can describe what happens to people who have the disease, but we don't understand the basic biology or biochemistry of what goes wrong. And I guarantee you that absence of knowledge is a huge stumbling block to the development of better therapies. Just imagine if you had to repair an automobile without understanding something about the mechanics of the carburetor. My wife pointed out to me that not everybody remembers what a carburetor is anymore. I suspect my son doesn't, but you know, I suspect I'm in good company here. You know, you just can't fix things that you don't understand. Final issue is that everybody in this room has a somewhat different inherent susceptibility to different diseases because of different genetic makeup. We are all different in our genomes. We have slightly different variants and all of that. This is why we look different and think different and have different susceptibility to disease. And that individual pattern of disease development is a very important stumbling block to understanding these diseases, as we'll see. And, as you may know, we all respond differently to drugs. And drugs that are sometimes very helpful for one person can actually make another one even sicker. And so better understanding those issues will really help us in the development of new therapies. So how can stem cells help? You know, what's all this hype or hope about? Well, let's start by defining a stem cell, because the newspapers and media have tried. Sometimes they get it and sometimes they don't. So remember that cells grow by dividing to make more cells. And all that a stem cell is a cell that can divide either to give rise to more stem cells, to perpetuate itself, or when treated or subjected to appropriate chemical or biochemical signaling molecules, will respond by differentiating, by specializing to take on one of those special jobs of cells we find in an adult. A neuron, a beta cell, a muscle cell, what have you. So stem cells are cells that can divide to give rise to either more stem cells or to the specialized cell types that we find in adults. Now, why is that useful? Well, there are a number of reasons. First of all, if you could really learn to control stem cells so that you could grow large quantities and then induce them to make beta cells missing in diabetes, cells of the substantia nigra missing in Parkinson's disease or what have you, they are potentially, in the long run, the raw material to replace damaged cells and organs. And that's a very hopeful thought. And I'll show you some evidence that that may indeed come to pass. Second, frequently less emphasized in the media is the fact that these cells, when they come from humans, will give us incredible tools to understand and thus better treat many of the diseases that we currently lack appropriate therapies for. And I'll show you some examples of that in a few minutes. And finally, they are very powerful tools as a result to better test drugs prior to putting them into humans and then into the human marketplace. And I'll show you some examples of that. And in fact, while this will seem morally callous given some of the debate about the ethics of this area of research, if we just step aside from the ethics for a moment and think strictly about science and medicine, they're actually what we sometimes refer to as an enabling technology. That is, it's a broad array of related methods that allow us to attack a number of otherwise unrelated problems and make progress. And in this case, that problem is human disease. Now, let's talk about some examples of naturally occurring stem cells that we encounter in normal human and animal biology. And of course, the first thing I'm going to do is tell you that the first cell I've diagrammed here is not really a stem cell. But, you know, what exceptions, huh? So there is, at the beginning of human and animal development, what's sometimes called a zygote. Forget about that. But what's most important is the first cell that starts development is what's referred to as a totipotent cell. An awful word. I didn't invent it. It's not my fault. But it's actually a pretty simple word to understand. So potent, that's the same root as potential. It means ability, right? Toti is the same root as total. All abilities. This is a cell that ultimately is capable of making every single cell type, or giving rise to every single cell type in the embryo, the fetus, and the adult. It's an amazing cell. Later in development, we encounter so-called pluripotent stem cells, which will lead, as we'll see, to embryonic stem cells in the laboratory. And these are cells that can replenish themselves, grow by dividing to make more, or, and more importantly, have this property of pluripotency, potent potential, pluri, the same root as plural, many potentials. These cells know how to make every single cell type in the adult, but don't know how to make all of the cells needed to start a pregnancy. They can't make the cells, the placenta or the cord or things like that. So they're more restricted, as we say, than these totipotent cells. And then finally, there are stem cells in adults, such as ourselves. There are stem cells in our bone marrow. There are stem cells in our liver, our skin, our brain, many of our tissues, perhaps not all. It's not so clear that there's a stem cell in the pancreas, for example, to make beta cells. But these stem cells that we find in adults, as near as we can tell from decades of work, are more restricted in their abilities than are these pluripotent stem cells that we find in the early embryos. So for example, stem cells in the bone marrow are largely capable of making cells of the blood and cells of the immune system. Stem cells in the liver are largely capable of making liver cells. And if you think about it, that's good business for the animal or the person. You don't want the stem cells in your bone marrow to start chunking out liver cells, or the cells in your liver to start making muscle, right? So you want them to be restricted in their abilities in their appropriate locations. And most data suggest that that is in fact true. We'll talk a little bit more about that in a few minutes because there are some potential exceptions, although I'm skeptical about their veracity. Okay, so what do we know about stem cells from adults? First of all, we know that they're enormously useful. In particular, if you have a disease of the blood or the immune system, such as leukemia or sickle cell disease, stem cells from bone marrow can be, in some cases, almost curative, enormously useful therapies. On the other hand, if you have type 1 diabetes or Parkinson's disease, we don't really know of a good type of adult stem cell to develop a treatment with. And we have maybe a few clues here and there, but we're really up against a wall in many ways. That's not to say that there aren't some ideas, but they don't necessarily look like they're going to go at least quickly. And that's because much of the work suggests that these stem cells from adults, as I just mentioned, have restricted potency and in many cases have restricted ability to grow. So for example, there has been an enormous investment in the pharmaceutical and biotech industries to learn how to grow large quantities of bone marrow stem cells. And it really hasn't been terribly successful. Hundreds of millions of dollars have gone into this, and it really hasn't been a winner. That's not to say that somebody's not going to figure it out at some point, but it hasn't been real fruitful. In fact, in some organs of our bodies, we know the stem cells are in there, but we don't always know who they are, in the sense that we can sort of see their footprints, but we haven't been able to find their little feet. And so there are, pretty serious identification issues as well that need to be solved. Not to say that they're not solvable, but they need to be worked on. They're not there. Now, I'll mention that there are some recent experiments, and you hear about them in the media, and they've become part of the political debate about stem cell biology and stem cell medicine, that suggests that stem cells from adults might be able to do everything that we want in medicine, that they can magically morph into any kind of cell. There are experiments that appeared on CNN a few years ago suggesting that stem cells from liposuction could make new neurons and new muscle, and in a nation such as ours, that was a very exciting development. But in fact, a lot of these experiments haven't been reproduced. Now remember, in science, you can go out on the street and say anything. But the gold standard is the ability for multiple laboratories to independently reproduce your work in order for it to be accepted as fact in our system. And second, you have to remember that science varies in quality also, as do most human endeavors. Some of these experiments are terrific. Some of them are poor. The newspaper doesn't always distinguish between a good experiment and a bad experiment from the standpoint of a scientist, I can tell you. And in some cases, they're just anecdotal in a sense, that what has unfortunately entered the political debate is that if you think that adult stem cells are the only ethically appropriate way to try to treat human disease with stem cells, my observation has been that these people sometimes will take some of the work on these cells out of context. And so you'll hear anecdotal things like, so-and-so in Portugal used adult stem cells to treat 20 people with spinal cord injury and they walked. Well, you have to remember that's not the same as an actual clinical trial. Clinical trial, you have a control group, you have enough patients that you know statistically that you're not just looking at random variation among the disease population, and then it has to be published in a peer-reviewed journal where everybody is free to look at the data and draw their own conclusions. And an awful lot of this stuff that you see injected into this debate, unfortunately, by non-scientists who have moral or political reasons for taking this view is just not borne out by scientific data. Unfortunate but true in the eyes of scientists. Now, I don't want to leave you with the impression that I think adult stem cells are not a terrific opportunity for understanding and treating human disease. They're wonderful. There's a lot of really great stuff to be done. But you have to remember that if you talk to our best adult stem cell scientists in the US and around the world, if you talk to the major scientific organizations, if you talk to the National Academy of Sciences, they're all going to say the same thing. Work on adult stem cells is terrific, but you should not walk away thinking that it can substitute, scientifically and medically, for work on other sources of stem cells that may be more ethically, shall we say, challenged. Now then, what do we know about some of these more ethically challenged cells? So let's talk about the properties of these pluripotent stem cells and then talk a little bit about where they come from and why scientists are so excited about them in spite of some of the ethical and moral issues that we face. Well, first of all, remember that these cells can grow to give rise to more of themselves. And in fact, under appropriate conditions, you can get a lot of them. I mean, we're talking liters, gallons, buckets, which if you want to have therapies deliverable to, more than just, a few mice, we're big animals. You're often going to have a fair quantity of stuff, or if you want to do more than one patient. Now, the reason these cells are so interesting, of course, is not only can they grow and divide to give rise to lots, but they can be induced to make heart muscle. That's been done. Fat cells. I know everybody says, why would you need fat? But actually for treating burn victims, I've been informed, it's really important to be able to get a layer of subcutaneous fat in. And that could be useful. Skin, liver, muscle, blood cells, and my personal favorite, brain cells of various sorts. So these cells can do this, and of course the challenge is learning to really control them. And that's an important research project that lots of groups are working on. Now, where do they come from? Where does the ethical challenge come from? And that's a source issue. Now remember that the way human and animal development starts is when a sperm cell meets an egg cell, otherwise known as an oocyte in my business, and those cells fuse to make a new cell, which is the zygote. And that zygote, as we said, is totipotent. Now, after a few days of growth and division, the zygote gives rise to this guy shown here called the blastocyst. Now, the blastocyst is the star of the national drama, the debates at the United Nations, and a number of electoral campaigns around the world. Now, it's really not all that striking to look at. It's about the size of a pinpoint on a piece of paper. You can barely see it with your eye without a microscope. It's a hollow ball, like a tennis ball, made of cells. It's got about 200 cells in it. It doesn't have any organs, no heart, no brain, no liver, no blood, no consciousness, no nothing. But it's enormously powerful because if the blastocyst is implanted, that is, placed into a human uterus, a woman's uterus, womb, in the case of humans, or in animals and theirs, right? It will fuse with the wall of the uterus and start a pregnancy, ultimately giving rise to a fetus and then an adult, a baby, a person. Well, I guess a baby's not really an adult. I actually understand that. But you know what I mean. Now, that's pretty exciting. Now, the issue becomes this. It is possible via a method known as in vitro fertilization, which is a medical procedure used to help couples who are having trouble having children the good old-fashioned way, to have kids. And what can be done is this fertilization step through the development of the blastocyst can be done in a laboratory, in a dish, and in a politically incorrect statement, with no women in attendance. You can take that where you want. Now, if that blastocyst that has been generated in a dish in the lab is implanted into a woman's uterus, it can, at a significant frequency, although not 100% of the time, as you know, give rise to a pregnancy, a fetus, and then a baby. Now, it turns out that in the course of in vitro fertilization treatments for infertility, not all of the blastocysts that are generated are implanted. And a couple reasons for that. Sometimes in the lab, when the fertility docs look at these blastocysts, they don't look very healthy. They don't implant them. Sometimes there are more generated than you should implant. So it turns out that the procedure for donating oocytes for this procedure, egg cells, requires some hyperovulation procedures and things that are uncomfortable. And so doctors try to generate more oocytes than you're going to need on that day. You generate a bunch of blastocysts. You don't want to implant 10 necessarily, because you don't want octuplets. Well, maybe some people do. I don't know. So there's always this balancing act between implanting enough to have the likelihood of getting a pregnancy high, while not implanting too many so that you get multiple births. And then the ones that are not implanted can be put in the freezer. And actually, they will stay viable in the freezer for 5 or 10 years sometimes. Now, the problem comes when couples sometimes decide not to have any more implanted, and they will, on occasion, decide to have them destroyed. Occasionally, they'll donate them to other people. That's actually quite rare. They'll keep them in the freezer indefinitely sometimes, and my experience with freezers is eventually the freezer breaks. Sometimes they'll donate them for research. Now, if they donate them for research, it is possible in humans, and it's also been, the original work, of course, was in animals, but recently in humans, to take these human blastocysts, extract the cells in the center, the so-called cells of the inner cell mass, and grow them in a dish in the laboratory. And those cells are remarkable because they will grow for long periods of time, and they retain, if they're treated properly, this property of pluripotency, the ability to make all these different cell types of the adult. And they'll do it in dishes sometimes. It's pretty amazing. Now, there's another source of pluripotent stem cells that has gotten everybody really wound up, politically and morally, because of its tangle with so-called cloning. So let's look at what that really is. And it's a method called nuclear transfer. Now, we're not talking about nuclear power. We're talking about nuclear transfer. And it's a biological procedure. And nuclear, in this case, refers to the part of the cell that has the genetic material, the DNA, that has all the instructions for running the cell and running the organism. It is possible, it turns out, from animals and more recently from humans, to take an egg cell, an oocyte, and physically remove its nucleus, remove its genetic material. And what you're left with, in the eyes of a biologist at least, is in a sense a bag of molecules that can deliver instructions to DNA, to genetic material. If you then take, for example, a skin cell, what we sometimes call a somatic cell, from a person, or if it's done in animals, obviously an animal, and remove its nucleus, its genetic material, and put it into that oocyte that doesn't have its own genetic material, you've now got a fused hybrid cell, and you can treat that cell with chemicals and electricity, and it will start to grow and divide and give rise in some cases to what looks for all the world like a blastocyst. Now then, you may be asking yourself, why would somebody want to do that? And of course, the answer is that those blastocysts can be used to generate cultured pluripotent stem cells. And if that starting cell, the donor here, was taken from a person that has a sporadic or a hereditary disease, these stem cells at the end are almost identical genetically to the cells of this person, which means that if you transplant them into that person, they may not be rejected as readily. Remember, one of the problems with organ transplantation is if I donate a kidney to somebody in this room, your body may decide that that's not genetically similar enough and decide it's an invader and get rid of it. That's not good for the transplant procedure. And you know of other examples of this, I'm sure. So this is a very powerful method potentially for that. Or if this person has a disease that you don't understand, you might be able to study versions of it in the dish that are genetically very similar to that person. And that'll become important in a few minutes. So that's why you would want to do that. Why wouldn't you want to do this? Well, you wouldn't want to do that is because if you were to implant that blastocyst into a human woman, or, as has been done in animals, if you were to implant it into an animal's uterus, you'd ultimately get a cloned animal or a cloned person. That would be bad. Our society has taken the view that we don't want to be cloning people. I'm not even going to comment on the ethical debate about cloning people. I'm just going to tell you that based on the data we have in animals, this is not safe. In fact, it's not likely to work in people. It barely works in animals. So you may know that there have been, lots of mice have been cloned in this way. A lot of cows, a couple of cats, some goats. I think a mule is out there. There's sort of a grab bag of animals. What's frequently not noticed is that most of the pregnancies that gave rise to those animals spontaneously aborted. Many of them threatened or killed the mother that carried them. And in the eyes of people working in this field, there's pretty good agreement that thus far, there is no evidence that any of the animals born in this way are normal. All right? So, on the face of it, this looks pretty unsafe. You wouldn't even begin to try this in people because it's patently so unsafe. And so, independent of what you think about the ethics of cloning for human reproduction, it's just not a safe thing to do. All credible scientists and doctors, in my view, agree that this is something we just shouldn't be doing in people. And in fact, in California law, it is illegal to do this in people. But many of us would like to preserve this ability to make stem cells for understanding and potentially treating disease. And that's where some of the political debate will come in a few minutes. Now, what do we really know about these pluripotent stem cells that are useful for thinking about science and medicine? Well, first of all, when I say they're pluripotent, I say that with very little doubt. And there's not much that I say this strongly. I don't trust almost anything. But this one is pretty solid. In the mouse, we have 20 years of data that establish that these embryonic stem cells in a dish are in fact pluripotent. It is a daily operation in laboratories such as mine, around the nation, around the world, there are dozens of them in San Diego, to demonstrate repeatedly that those cells are pluripotent. This is something we actually know. Now in the human embryonic stem cells, the evidence is not as strong. It's not as strong because the sort of experiment you can do in the mouse where the cells are implanted and things like that, it's a little bit more ethically tricky to do in people. I mean, experimenting with people is a much dicier business for obvious reasons. But the evidence that exists for how these cells behave in the lab in a dish really supports the notion that they, the human cells, like their mouse counterparts, are in fact pluripotent. As a result of that pluripotency, there's a very solid experimental and theoretical foundation to start inducing the development of specialized cells and ultimately to solve some of our problems of tissue or organ rejection during transplant. I mentioned they can have good growth properties, and I'll just state there's a lot of very promising proof of concept or proof of principle research in animal models of disease with these pluripotent stem cells from people. Beautiful data developing on spinal cord injury in animal models, mice and rats, Parkinson's disease models, some work on diabetes that looks reasonably promising, and a number of others. So there's good evidence to believe that this line of research, if pursued, based on what we've seen thus far in animals, is going to lead us somewhere very powerful in people. Okay, so let me give you a couple of specific examples about how these cells might be and are beginning to be useful for human therapy development. First is toxicity testing. Now remember, when we were talking about the development of new therapies, we mentioned that sometimes things that get tested in animals, you put them into human clinical trials and all of a sudden you discover they're toxic to the liver in humans, whereas they weren't in mice or dogs or something like that. Well, one of the things that's very promising about this technology is it may be possible to generate large quantities of defined liver cells from these pluripotent stem cell lines in the laboratory and use that directly for initial toxicity testing prior to putting the drugs into people. And this could turn out to be extremely powerful for helping us prioritize and get rid of those things that are going to turn out to be toxic before you spend $50 or $100 million on the first parts of a clinical trial. That's pretty good business in my view, and will help us with the development of drugs. Similarly, we may be able to make kidney cells or other cells like this for testing. Maybe a second example. One of the problems that my own laboratory works on is Alzheimer's disease. Now, you probably know that we can describe pretty well what happens in Alzheimer's disease, right? People start forgetting things, they don't recognize things as well, and eventually many of the cells in the brain die. And there's a set of pathologies that go along with it. We can describe that pretty well. We don't really know why it happens. And we don't quite know why those brain cells fail to function properly and why they die. So how do you figure a problem like that out? Just think about that for a minute. It's possible, though not likely, that many of you in this room are prepared to step forward and donate your brains to my experiments right now. However, it is unlikely. That's right. You have other uses in mind for them at the moment. So where are scientists, such as myself and my laboratory, going to get human brain cells in the earliest stages of disease to try to understand what's going wrong? So one mechanism, one thing we do, is we know that in humans there are rare forms of Alzheimer's disease that are hereditary. And the genes that mutate to cause those diseases have been identified. And you can put those mutant genes in animals such as mice, and the mice get a disease that looks something like Alzheimer's disease. And you can learn a lot about what goes wrong there. Problem is that the mice don't really get Alzheimer's disease. They have some of the symptoms, but not all. And so you can use those animals to develop ideas, to develop hypotheses for what's going on, and maybe even develop some drugs. But now we got a problem again. How do we test this in people? Well, clinical trials is one way, but that's a pretty expensive way to test a hypothesis. And so what my lab is currently doing is to take another approach to this, which is to take these cultured human pluripotent stem cells and then to introduce into those cells, into those human cells, the mutations that cause Alzheimer's disease in some people. And what that will give us are human brain cells in dishes in the laboratory that have those mutations that cause Alzheimer's disease. And we can use those cells to test our ideas about disease that come from animals, that come from mice. And in addition, we can track the early stages of their biochemistry and physiology, and that will let us both test hypotheses for disease generation, and obviously for hypotheses that turn out to be correct, will help us then test drugs for the treatment, and perhaps develop new drugs for the treatment of a disease which I don't need to remind you, we have almost no therapies for. We got virtually nothing. Now, that's of course for these rare hereditary forms of Alzheimer's disease, but what you also know is that most Alzheimer's disease is what we call sporadic. It doesn't have obvious, doesn't get passed from parents to child to the next generation, so obviously. But we do know that there is some genetic component to it. We're not quite sure how much of it is there and exactly how it works. And this is where I think the nuclear transfer method is going to help us a lot, because it will enable us to potentially get brain cells derived from people who have sporadic Alzheimer's disease and test their biochemistry using this nuclear transfer method I mentioned to make cloned stem cells, genetically identical stem cells to somebody who has disease, and ask, are the biochemical changes the same as what we see in this known hereditary form? Do they respond to drugs in the same way? Do they have the same physiological changes? Because otherwise, we don't have mice that get sporadic Alzheimer's disease. We got nothing there. So what are the social issues that play out here? Well, some of them are obvious, right? This is the understatement of the century, and it's only 4 years old, The controversial moral status of the human blastocyst, or embryo or pre-embryo, depending on whose definition you're using. But it really comes right down to that ball of cells. And I can tell you that I have debated Catholic theologians, other people. I call it a blastocyst, they call it a baby. And they believe it. And some people do look at it that way. And so if you look at that ball of cells in a dish or in a freezer as being equivalent to one of us in this room, then of course we may not use that for research. I mean, that's a legitimate point of view. If, however, you look at that ball of cells as I do, as not being equivalent to one of my kids, and believing as I do that doing research with cells from those will be very beneficial to the treatment of human disease someday, then you must work with them. Because that is one of the major goals of biomedical research, and that's what we do. We try to understand basic biological principles and then apply that to human disease. They are indeed conflicting moral imperatives. Now I'll just point out that first of all, American religious and ethical viewpoints are pretty diverse on this issue. They range from, as I mentioned, baby on one hand to just ball of cells on the other and everything in between. And I'm sure that if we went through this room of 50 people, we'd find 75 views on this. But I'll just stress, moral and ethical people of good conscience will disagree. I mean, I have on occasion been compared to the Nazis. I don't like that. I think that's a little over the top, but people disagree about this. But I will ask this question. Is it the case that most Americans think that this hollow ball of 200 or so cells that has no organs and which is growing in a dish, or even more is frozen in a freezer and can be revived? Do we really think of that as being equivalent to one of us in this room or one of our kids or something like that? I actually, based on my travels over the past few years, don't think that most people really think that. I think there's a much broader gradation of opinion, or belief, or philosophy, or whatever it is for different people. The second social issue is public antipathy towards cloning, right? This is a sort of a funny issue in a sense, because what tends to happen in non-scientific groups is I say cloning, you think of armies of Arnold Schwarzeneggers terrorizing the countryside, right? Did I get it right? But let's remember something here. We love cloning, in fact, right? Remember what cloning is? It's a verb. It means to make a copy of something, a replica, make a replica, right? We clone software. We clone DNA. We clone human DNA all the time to make human insulin, to make human erythropoietin, to make human drugs. It's the basis of the entire pharmaceutical, biotech, and biomedical research enterprises in our state and our nation. We clone cells to study cancer treatments. We clone viruses to make vaccines. What we don't do, the one thing we agree on, is we don't want to clone people. It's illegal to clone people in the state of California. I don't want to clone people. My colleagues don't want to clone people. But we do want to be able to clone cells, DNA, viruses, and all the rest of this stuff to do the kind of research we want to do. Now, I'll just say that opponents of this sort of research out there will say, these guys just want to, it's all about human cloning. Cloning. And I kind of resent that, because that's not really true. I think you have to be specific. It's not as indefinite as all that. The problem of language. Need I say more? How you talk about things are everything. And it's part of the problem. I say embryo, and somebody draws a picture of a little baby crawling out of a test tube. I mean, it's really all over the map. And so it's why sometimes I try to be precise. I talk about a blastocyst. I talk about cloning DNA or cloning cells or cloning a person. Let's be specific here. And if we're going to write law, let's really be specific when we write law. Because I can tell you, if you're not specific when you write law, you can really make a mess. Now, let me just stress something that is not so obvious, because part of the debate has been about, what kind of research should we even be doing? Part of the debate in the past few years has been, when is it appropriate for the public to fund research? And why is it appropriate for the public to fund research? And I think you have to remember a few things that are maybe not so evident. First of all, there are policy issues. When the public funds research projects, the public has a lot more information about what's going on, as well as control over how that research is conducted. Believe me, when research is paid for in private companies with venture capital or private sources of funds, it is often secret. We often do not know it, do not know what is going on, and that is appropriate, because companies are competing with other companies to develop products more rapidly, and secrecy is an appropriate thing in industry. But when the public is involved, when it funds research in institutions such as this one, most of it is pretty open, with rare exception. It's published in the scientific literature, the public hears about it, we talk about it in open fora, such as this one, and you get a lot more input into what happens. Now, the second issue is that there are enormous financial and business limits in the private sector, and we'll talk about those in a moment. But in fact, for the past 50 years, the public has been the early stage funders of basic biomedical research that ultimately leads to therapies, treatments, and understanding of disease. And third, public institutions such as this one, and Stanford and Salk, which are effectively public institutions, they're largely funded with taxpayer money. Many of our best scientists are in these institutions in this country. You want an area of science to move quickly, you want your best guys involved. And that's how you get it. Now let's talk a little bit about what the financial pipeline looks like to new therapies, because people don't always realize how it actually works. And we can debate about how it should work, but let's talk about how it really does work and worry about how you might change it in a different topic. But it's worked this way for the past 50 years, and I don't see a quick fix coming. Remember that basic research ultimately leads to ideas, discovery and validation, which then lead to therapy development, clinical trials, and then ultimately approval and marketing, right? Actually a drug on the market or a device or a therapy that you can use and procure for people. Doesn't do anybody in this room any good if there's a great idea in a lab that doesn't find its way into a hospital or a company or someplace where you have access to it. Now, if you were to plot time and dollars, not surprisingly, early stage stuff is much, much cheaper than late stage stuff, right? In fact, it turns out that the basic research that really drives this whole thing is relatively inexpensive compared to, for example, the cost of clinical trials, which, as we just said, can run into hundreds of millions of dollars in many cases. Now, the way this is done in this country is, in fact, these early stages, particularly the earliest stages, are largely funded by philanthropy, which is not a very big fraction, by the way, and the federal government through the National Institutes of Health, the USDA, the Department of Energy, the Department of Defense. They all fund a lot of this early stage biomedical research with its goal as understanding basic biology and then translating that into therapies. Where you tend to see venture capital move in is later in this curve. And actually, there's not a huge amount of venture capital money out there. There's some, but it's not huge compared to the public investments, if I've got my numbers right. And it comes in relatively later when there's actually an idea about how a basic discovery or idea can be translated to a specific therapy or a specific product, where things like market size can be evaluated. Remember, venture capitalists are not the same as philanthropists. They must make a profit, right? Well, and let's think about it. Where do venture capitalists get their money? They're often investing money that are given to them by people in this room, right? They're fund investors. They're investing money that is given to them by somebody else with the hope for a return. They better make a profit or they're out of business and nobody's going to give them money to invest anymore, right? So they tend to come in later when there's more specific understanding of what's going to happen. And then the late stages, particularly when it gets expensive for clinical trials, a lot of that is done by the pharmaceutical industry. Some clinical trials are done federally, not enough in my view, but there are some. A lot of them are done by the major drug companies. And remember that revenue, actual money coming back into the system, none of this stuff makes money. You don't start getting money until out here, 5 to 10 years into that pipeline, and sometimes more, depending on how hard a therapy it was to develop. Now, this is not the only way to develop new therapies. This is the way that things go through the private sector. Some therapies, of course, are developed entirely within academic health centers. Organ transplant is a good example of that. There's not really a private sector route for that, right? There's no companies that sell kidneys or do kidney transplants or what have you. So, what happens when the federal government refuses, for political reasons in this case, to fund early stage research in an important area like embryonic stem cell research? It's not that these pieces move. You end up with a gap. You end up with a hole in the way it usually works. And as I say, you can argue about whether or not you want it to work this way. This is the system that has been established and actually works in some ways pretty well. But you can't just all of a sudden decide politically that, oh, we're not going to do that anymore and expect things to happen. It's just not reasonable. All right. So scientifically, of course, the need for research with human cells with public funding, of course, is that we need basic as well as applied. Companies excel at applied research, not so much at basic. We talked about why human and animal cells may not behave the same. And of course, if I'm looking at developing a therapy for ALS, which I'd like to try to do, I don't know that I'll be successful. I have some choices here. I could first get all the problems worked out with mouse cells and test them in the mouse, and then go to the human cells and try to work out all the problems in the human cells and apply them to the mouse and then to the human. Or I could skip a step and just start with the human cells, get the problems worked out, test them in the mouse, and then move to people. In my view, it makes a lot more sense to try to skip that intermediate step. No guarantee, but to me that makes sense. What are the legal issues? Well, in the midst of all this debate about whether cells from these blastocysts should be used in publicly funded research, abortion is legal in this country. Not everybody likes that, but it is legal. And I'll note that the destruction of blastocysts in IVF clinics in this country happens on a daily basis. They're created and destroyed by the hundreds, perhaps, thousands. I don't know the numbers. Nobody really does. It's legal, it's common, and it's okay as long as you're not using public funds. Go figure. The legislative situation. Well, there's a big debate about what research public funds should pay for, and there's a lot of confusing legislation in the Congress. The president has far less power here than he's given credit for. And I won't bore you with the details of the language. I think I'll just say that there is current legislation that makes it next to impossible to make new cell lines with federal dollars, and that's not the president's problem. It's a congressional problem. And second, I'll mention that every year for the past several years, bills have been pending and passed in the House, but not in the Senate, that would criminalize nuclear transfer. It would put me in jail if I did it. If a therapy was developed in Britain and you went and got it and put it in your body and came back, as we understand the way that legislation is written, you would be a criminal when you cross the border. There are people who object to this pretty strongly, and I understand why. I don't think these are the appropriate remedies. But that's just one man's opinion. Okay, now you may recall that when President Bush, while he was still candidate Bush, he said there was no way he was going to allow federal dollars to be used for research with these human embryonic stem cells. Adult stem cells, fine. Human embryonic stem cells, no. On August 9th, 2001, he announced that he had changed his position and announced that federal funds could be used for research with human embryonic stem cells. He would provide robust funding for both. He actually doesn't have that much control, it turns out, but that's fine. And there were a number of restrictions on what you could do with human embryonic stem cells. Every restriction was appropriate, save one, in my view. Most of them had to do with informed consent and protection of patients and donors and all the rest. Good standard stuff. Actually not as strong as I would have liked, it turns out. The requirements under his policies were less stringent than the Clinton ones, interestingly enough. But the kicker was this one. You could only use cell lines that were taken from blastocysts prior to August 9th, 2001 at 9 p.m. Eastern Standard Time. So a cell line that was taken from a blastocyst on August 10th was not eligible for funding with federal funds. Any of that $30 billion of NIH money couldn't be used for research with those cells. Well, that's sort of interesting. So what do we think about that plan? Well, there was good news. Publicly funded research has started. And on August 9th, 2001, the president made it okay to use public dollars for every stem cell line that was currently in existence. That was a good thing. All right? But there were questions. Were all the lines that he announced were available, were they of good quality? Could you really get them? Were they really there? Would they support therapeutic development? Were they good for more than just research? And then, of course, the big question was, if on August 10th, 11th, 12th, some really great lines, better than the original ones, became available, then what? And I think, the answer has become somewhat obvious. 3 years later, the policy is showing its age already. All right? So, in fact, although 60 to 80 lines were thought to be available on August 9, 2001, you're looking probably at 19 or 20. They're genetically not particularly diverse. That's an issue. And there are real questions about quality. They probably cannot be used in any clinical trials. There's evidence emerging that they're not as good as some of the more recent lines for making certain kinds of cells. And you know what? As a scientist, it was sort of always obvious that this was going to happen. Just think about it. Let's think about software. You got the first release, the second release. Let's think about the Edsel, then the the Wright brothers, then jets. The first time we do something, we don't always do it the best possible way for all time. In fact, you wouldn't expect us to. We do improve technology as we go. And that's the issue now. So the issues to think about, well, all right. So the slippery slope. Everybody likes to talk about the slippery slope, right? If we allow this, then all these terrible things might happen, or will happen, right? It's the notion that if we have the ability to do something wrong, we will therefore be driven to do it. My friend Paul Berg, who won a Nobel Prize for his work in recombinant DNA and who was part of the original recombinant DNA debates, likes to say, well, he'd like to see what the slope looks like and get there before he decides whether it's slippery or not. I think that's actually rather sensible. I would approach it in a different way and point out that every human technology that has ever been invented, this is pointed out to me by another friend of mine, Matt Meselson, every human technology has been misused. Fire, the automobile, I can take a hammer and use it to build a building or bash somebody's head in. Banning technology that has benefit, has utility, just because it might be misused, man, we'd still be back in caves and cold. If we took that view. A wise society does not do that, in my view. A wise society restricts the use of technology, not its development. It doesn't make any sense. That's because we need to learn how to balance potential benefits and risks. There's always risk. There's risk to proceeding. There's risk to not proceeding. And we have to do appropriate risk-benefit analyses. Not always so easy in the political world, I have to say. Criminalizing research methods versus withholding public funding. Obviously, it's one thing to say we're not going to fund you. It's another thing to say we're going to throw you in jail if you do it. The thresholds ought to be different there. And I think the current situation is just goofy on this. Finally, we need a lot more data before we make final decisions. I have to say that, again, as a scientist, decisions to prohibit progress, in my view, those should have the highest standard of evidence to drive them, not the lowest. It's one thing to say we need more data. Yeah, boy, we do. We got to do the work to find those data. To say that we're going to stop the research before you can even get the data, and oh, by the way, you got to prove that it's going to be good before we let you go on. That's a hard one in my view. I'm quite biased about that. I just don't understand that. The issue for a society is, where do you draw the line on what you do and what you don't do? For nuclear transfer, of course, there are people who would make that act criminal, right? Throw you in jail for doing that. I think that's bad. California, happily, has by law put the line right there. You may make stem cells, you may not make babies, or even try. Okay, that makes sense to me. Orrin Hatch, a noted conservative Republican, he's actually agreed with the California approach. He's at odds with the president on this one. and certainly at odds with his colleagues in the Senate and the House. The AMA has drawn the line here, as have many medical and patient groups and scientific groups and everything else. There's pretty broad consensus that we shouldn't do this. There's controversy about whether we should do this or not. And again, if you're going to ban something, we ought to at least ban the thing that we agree on, but not the thing that we're disagreeing about. Again, that doesn't make sense. Okay, predicting the near future. Oh boy. Presidential policy change is unlikely. Well, that's clear. I think that federal legislation is unlikely before Election Day, particularly with it drawing close, and it's unlikely after, I have to say, for a variety of reasons. There's a very complex research landscape. I can just tell you as a scientist trying to operate and get research that I believe is potentially valuable and beneficial moving, given this sort of political quagmire and funding quagmire is, shall we say, complicated. We really lack a broad national consensus. That's one of the problems, I think. Our ability to forge a national consensus has really been hindered by lack of information. It's one of the reasons I think it's so important for groups like yours to take the time to inform yourselves, because the only way to counter misinformation is with understanding and better information. And my detractors, I think, don't always use accurate language and data. Are the states appropriate to move ahead? Well, that seems to be what's happening. I mean, in the absence of a federal approach, you're starting to see state approaches spring up. California's gone one way, Arkansas is going another way, Washington State is thinking about which way it's going to go. We may end up with a patchwork. That does allow more latitude for disagreement. In fact, the framers said, the states were the laboratory of experimentation. Maybe that's good, I don't know. It has benefits and problems, if it's okay to do it in California but not in Arizona, I'm not sure how I think about that, but that may be what we end up with. It will allow progress, but there'll be some limits. Finally, California, we'll see what happens. So thank you for your attention. I'd be happy to take some questions.